This is the presentation of the journal article for Cell Biology for Jake Thompson. So my article is The Structure and Function of Insulin, Decoding the TR Transition by Michael A. Weiss. And this is the front, and what it talks about is the insulin hexamer with the zinc 2 ions, and it talks about how with mutations inside of it, it can actually cause type 1 diabetes, and how when we look at crystallizations, it can show what there is wrong with it. This first section of the introduction, it reiterates what I said in my last video. It shows insulin having 2 chains, an A chain and a B chain. The A chain has 21 sections and the B chain has 30. And it talks about how when it's stored in the beta cell, it has a zinc 2 plus ion stabilizing hexamer. And the hexamer is released as a monomer with the hormone in the bloodstream. This is the diagram showing the release of the zinc 2 plus crystals into the bloodstream as monomers. And it says right here up at the top that we do not know how insulin binds to the receptor completely. This figure right here shows the 3 common hexamers of insulin. There's T6, T3R3F, and R6. And the assembly of these hexamers by the A chain and the B chain of insulin to make the hexamers. This page shows an example of crystallography that is used for the insulin hexamers. And it also talks about how the folding of proinsulin in the ER actually helps with the hormone usage whenever it binds to the IR, which the IR is insulin resistance, which raises your blood sugar level. The second section takes a look at the mutagenesis of insulin. It talks about kind of a beta turn between the B7 and B10 parts of the B chain. One important thing that it talks about a little bit more in the next part of the chapter is about glycine at beta 8. Whenever there's a substitution at this 8th location, it can impair folding inside of the ER. And it shows whenever you place it with 1 proteins and D proteins, it actually can prevent certain crucial parts in the process. So on this page, it shows whenever there is the substitution at the beta 8, usually what it does is it prevents the disulfide bonds that I talked about in the last video from actually occurring and making a full structure of the protein. The next chapter shows whenever you replace the glycine with 1 serine, even though it can prevent other crucial functions inside of it, it can actually have more flexibility. And whenever you replace it with D-serine, it can prevent the disulfide pairing, but it can still have a positive effect for the insulin. This third section just talks about how there's a collection of mutations that cause diabetes. The most common one is when you remove cysteine from proinsulin, which gives you an odd number of thiol groups. This causes a misfolding in the ER. And whenever there isn't one that directly involves cysteine, you can have hotspots on B5 and B8. B8 being on the B chain that I talked about earlier, involving the glycine. And the last page just summarizes everything they talked about earlier in the article. For my paper, I would like to use how the insulin functions as a hormone, as a hexamer, and how we study it and use it to our advantage.